Listen only mode.
Good morning, good afternoon, good evening. Welcome to our first research webinar. The Phelan-McDermid Syndrome Foundation is pleased to organize the first of what we hope will be a series of webinars related to Phelan-McDermid Syndrome, research in medicine. My name is Sue Lomas. I'm the President and Executive Director of the Phelan-McDermid Syndrome Foundation. I live in Venice, Florida with my husband and 17-year-old son who has Phelan-McDermid Syndrome. It is also the home of the office of the foundation. We are recording the session today, so there'll be a chance to view it again. Attendees will be muted during the webinar. Once we start the discussion, we'll post instructions for asking questions. We'll address as many as possible at the end of the webinar. Any questions that we do not have time to address during the webinar will be addressed in a follow-up email. If you are viewing the webinar online and have audio problems, please make sure that your volume is turned up. You can also listen to the webinar by phone by calling 415-363-0079. The access code is 445-895-705. I'd like to now turn it over to our amazing research chair and today's interviewer, Geraldine.
Thank you, Sue. I'm going to repeat that phone number in case you're having audio problems. It's 415-363-0079, and the access code is 445-895-705. My name is Geraldine Bliss, and I chair the Research Support Committee of the Phelan-McDermid Syndrome Foundation. I live in Houston with my husband and two sons. My 13-year-old has Phelan-McDermid Syndrome. Today we are joined by Dr. Joseph Buxbaum and Dr. Alex Kolobzon, investigators at the Seaver Autism Center at Mount Sinai School of Medicine in New York City. Dr. Buxbaum is a professor of psychiatry, neuroscience, and genetics and genomic sciences at the Mount Sinai School of Medicine, where he is also director of the Seaver Autism Center for Research and Treatment. He is a world-renowned molecular geneticist and spearheads research into human psychiatric and neurological diseases. As Vice Chair for Research in Psychiatry, Dr. Buxbaum helped set the research, which is ranked among the top 20 psychiatry departments in the country in NIH funding. In the area of autism, Dr. Buxbaum and his colleagues are employing techniques of molecular genetics to identify and ultimately characterize genes that contribute to autism susceptibility. His laboratory has taken the findings regarding the causes of autism and translated them into animal models where therapeutic approaches can be evaluated. In this context, Dr. Buxbaum has established the Autism Model Systems Initiative, which makes use of multiple experimental systems to develop and evaluate novel therapeutics in autism spectrum conditions. Dr. Buxbaum is also part of several large consortia that are focused on this area of research. As part of his role in the Seaver Autism Center, Dr. Buxbaum oversees clinical studies evaluating therapeutic options for individuals with autism spectrum disorders. He also serves as co-editor-in-chief of the journal Molecular Autism. In recognition of his scientific contributions, Dr. Buxbaum has received numerous awards, most recently the Mount Sinai 2011 Dean's Award for Excellence in Translational Science and the Joel Elkes International Award from the American College of Neuropsychopharmacology. Dr. Buxbaum received his PhD from the Weizmann Institute of Science in Rehovot, Israel, and was a postdoctoral fellow at the Rockefeller University. Dr. Kolobzon is a child and adolescent psychiatrist and an associate professor of psychiatry and pediatrics at the Mount Sinai School of Medicine. Dr. Kolobzon completed residency and fellowship training at Mount Sinai. He was the director of clinical services in the Division of Child and Adolescent Psychiatry before joining the Seaver Autism Center as clinical director in 2007. Dr. Kolobzon has published numerous papers on autism and has clinical expertise in the psychopharmacology of autism and related conditions. His research is focused on developing novel pharmacological treatments, and he has significant experience as a principal and co-investigator on many clinical trials. Most recently, Dr. Kolobzon has focused on studying patients with Phelan-McDermid syndrome in an effort to better understand the clinical presentation and has collaborated closely with basic scientists to explore possible targets for pharmacological intervention. He is also extremely committed to medical student and residency training and education. He is the Associate Residency Training Director in Child and Adolescent Psychiatry and co-director of the Dietrich Hamburg Medical Student Training Fellowship in Child and Adolescent Psychiatry, sponsored by the Klingenstein Third Generation Foundation. He has written 3 books designed for medical student and resident education and co-edited the textbook of Autism Spectrum Disorders published by the American Psychiatric Press. He is an active teacher, mentor, and clinical supervisor and has reached many teaching awards as well as a grant to support innovative educational endeavors. Welcome, Dr. Buxbaum and Dr. Kolovzon. Thank you so much for your participation in the Phelan-McDermid Syndrome Foundation's first webinar. We're really pleased to have your participation today.
Thanks, Caroline. It's a pleasure.
Today's discussion will begin with Dr. Buxbaum's basic findings and how they are being translated into a potential new therapeutic for Phelan-McDermid syndrome. Dr. Kolovzon will discuss the findings from his clinical research and what the next steps will be. We hope to have about 5 or 10 minutes at the end for questions. If you'd like to ask a question, please use the messaging system on the GoToWebinar panel on your computer screen. Okay, Dr. Buxbaum, let's back up a few years. What led to your research interest in Phelan-McDermid syndrome?
So, you know, I'll give you two answers. The first one is that we now know that mutations in Shank 3 are one of the more common causes of developmental disorders, autism spectrum and otherwise. People think about Fragile X as being perhaps the most common monogenic form of autism or developmental delay. Shank 3 deletions are almost as prevalent. So that's one reason to study it. It's also a gene that's very interesting because it lies at the heart of the connection between nerve cells and the brain, and that pathway is probably altered in many sorts of developmental disorders that involve behavior or cognition.
Thank you. We hear a lot about the importance of mouse models in autism research. Can you briefly explain why they are so important?
Well, there are 2 facets to that. The first is that there's technology now to modify any gene in a mouse, and there's emerging technology to do the same thing in rats. So that means if you have a human disorder caused by a mutation in a gene, you can mutate the same gene in a mouse. So you're able to capture the same biological deficit. And then the obvious follow-up there is that mice are much more tractable as study subjects than humans. So not to make light of it, but there are things we can do in a mouse in terms of studying brain function that would not be possible in humans. And we can also screen drugs in mice in a preclinical space to find ones that work in your mouse model. Once you find ones that are both effective and safe, can be translated into the human population.
Right. Okay. So you were the first to publish findings from a Shank 3 knockout mouse. Tell us what you learned from that particular mouse model.
So we looked at several measures of kind of brain functioning in the mice. The first level was almost the most basic level. We asked how nerve cells communicate, how effectively they communicate in the brain of the mice. And what we found was that there was a pretty subtle deficit in the connection between 2 nerve cells, both in terms of the kind of baseline connection, but also in terms of how the nerves change their properties in response to learning and memory. So it told us about, if you will, a cellular deficit that was changed when you delete Shank 3. And the cellular deficit, or the nerve cell deficit, seemed to map out onto a specific receptor and part of the cell, which again kind of led us to think more about what therapeutics would be most useful to intervene in the deficit that we saw. We also saw some very subtle behavioral deficits, but those are turning out to be very important because when we try drugs, we can try the electrophysiological study, which is very involved, or we can try to look at the behavioral changes and then see what drugs reverse that. And in fact, what we do right now is we do both. We look at both changes in nerve function as well as changes in behavior in the mice.
Okay. So how were your findings from your mouse model relevant to PMS in particular and to autism more generally?
So in terms of, but there's a lot of, I mean, from my perspective, well, let's take PMS first. PMS is characterized by some cognitive deficits, by some perhaps autism-like traits, well, certainly autism-like traits in about 75% of the kids, some motor deficits, and in the mice, you know, we see some similar features. We see the motor deficits, we see some changes in molecular and cellular traces of learning and memory, and so insofar as a mouse reflects human things, and it's certainly not perfect, we see a lot of the same kinds of changes in the mice that you would see in individuals with PMS. And that tells us that the mice have what we call face validity. They kind of reflect what we're looking for as endpoints that we want to improve with drugs. Now, in terms of autism, you know, there is a tremendous overlap between the features of autism and Phelan-McDermid syndrome. So we think that these mice are also going to be they're going to certainly be valuable for those subtypes of autism caused by either mutation in Shank3 or mutation in genes in that pathway. But we actually think that these mice will be valuable to understand much of autism. And I think that's the reason why, you know, the number of grants on Shank3 have gone from just one or two a few years ago to many, many dozens of applications, because people realize that this is really, in autism, one of the most significant new directions.
Right. We're really pleased. Yeah, of course, we're really pleased to see that kind of research energy around Shank3 and hope that it will mean good things for Phelan-McDermid syndrome. Dr. Kolovzon, around the time that Dr. Buxbaum's research was really starting to heat up, I know you were beginning to see some patients with Phelan-McDermid syndrome. How many patients have you now seen in clinic?
So we've now gotten to know about 32 families, actually, and seen them over the course of 2, 3, sometimes 4 days through pretty comprehensive evaluations. We've seen kids as young as under 2, and then we've even seen a few adults. People have come from the tri-state area, certainly, but also from Florida and Alabama and California and Washington and Chicago, and it's really been terrific to learn and to get to know these families and kids.
Great. Well, tell us about some of the most interesting or important observations about the patients with Phelan-McDermid syndrome and whether any of these findings were surprising to you.
So one of the things that we were focused on from the beginning was trying to understand where Phelan-McDermid syndrome fits in with autism and trying to get a sense of how autism was presenting in these kids. So a lot of our exams and evaluations were focused on autism symptoms, language impairment, repetitive behaviors, and social impairment. But we were also really interested in trying to find more objective ways of assessing these kids beyond just behavior. So things like motor skills were interesting to us, other kinds of neurological findings, gait, things like trying to think about language in a way that we could actually measure it objectively. These were all the kinds of things that we were targeting early on. Other things that we measured were around adaptive behavior, social functioning in general, fine and gross motor skills. And we weren't really expecting anything in particular. We were just setting out to learn, but I think most of us were surprised at how many people seemed to meet criteria for autism based on our current gold standard assessments. And I mean, we can talk about it in a little bit more specifically, but there are some issues with these assessments in terms of trying to evaluate kids with developmental disability that puts them at a mental age below 18 months, but nonetheless, about 50% of the kids that we met really full, strict criteria for autism using our assessments, and another 33% were also on the autism spectrum without meeting full criteria. And actually those who didn't meet full criteria were kids that had less repetitive behaviors than you would typically see in autism. They had the social impairment clearly, they had the language impairment clearly, but they didn't have the same degree of repetitive behaviors that you would otherwise require to meet full criteria for autism.
I was really surprised when you presented your findings at a poster at our symposium in March, and you presented this finding that over 80% meet criteria for autism spectrum disorders. I was really surprised. Were you surprised? Did you expect that the penetrance of autism would be so high in this community?
You know, so there have been about 150 cases described pretty well in the literature from 7 or 8 different series, and the rates are variable. Some reports and very small reports that do a limited evaluation will say that 0 kids meet criteria for autism. And then depending on the types of assessments and instruments that are being used, other reports will say all the way up to 94%. So I think this is an important area of study, and we didn't really know what exactly to expect. One thing that we're quite confident in was that the instruments that we use are they're absolutely the standard of care. I think we can take it a step further in the future in terms of better refining them to be more appropriate for kids who are more severely developmentally delayed. But nonetheless, this proportion of about 80% is somewhere in between if you look at the different studies and the varying rates. So was I surprised? I guess I was a little bit surprised, but I was also relieved. I think this is an important issue to be able to tie, I mean, for the foundation and for the families in general, to be able to tie Phelan-McDermid syndrome closely to autism is a valuable thing in terms of services, educational accommodations, in terms of research funding also. So in that sense, it was a very good finding for us.
I know that you see many other children besides just patients with Phelan-McDermid syndrome. You also see patients with Fragile X and tuberous sclerosis and many other autism-associated genetic conditions. And I think the last I read, the penetrance of autism in those communities is actually lower than what you're reporting in the community of Phelan-McDermid syndrome patients. Can you talk a little bit about the penetrance of autism in some of these other genetic conditions?
That's right. I mean, and Joe mentioned Fragile X syndrome, which has historically really been the classic model of a monogenic cause of autism. So in Fragile X syndrome, for example, the penetrance, as you say, is something like 40%. Other estimates may be lower, around 25%, but certainly 80% is very, very high.
Well, okay. Going back to some of the findings, the sort of the phenotypic findings, can you tell us about some of the maybe the more significant ones that you noticed? I know you, in the past I've heard you talk about some of the gait issues as a little bit of a surprising finding. Tell us more about what you saw with that and maybe some of the other kinds of findings and really what that means for treatment and for designing studies and looking at those particular characteristics as possible biomarkers or endpoints for clinical trials?
Sure, and I mean it's a really good question because historically, certainly in the autism literature, when you're talking about a behavioral syndrome like autism, it's been very challenging for us to try to figure out ways of measuring improvement in an objective way that can minimize the effects of placebo. So when you do a controlled trial and you're comparing an active medication to a placebo, and you're seeing families that are highly motivated and clinicians that are obviously highly motivated, there's a real bias towards kind of inflating this sense of improvement even if kids are on placebo, especially when you're talking about short-term trials. Over the course of 12, 16 weeks, it's not unusual for there to be 30, even 40% response rates among kids who are on placebo. And so in this particular trial, as we're moving forward, we're thinking very carefully of ways of trying to measure improvement using objective measures. And so in terms of the assessments that we've done, and when you mentioned gait's a good example of that in a sense, because you have this very clear change in gait among kids with Phelan-McDermid syndrome as compared to typically developing kids. It's not one specific kind of gait, actually. You see a lot of different kinds of qualities to the gait in these kids, but somewhere like, I mean, among the kids that we really looked at carefully, I think 15 out of 16 of them, for example, had clear gait disturbances. They had a wide-based stance, they had in-toeing as they would walk, there's some kids who were doing toe walking, a lot of them had something called steppage, which is sort of a high stepping gait, and these kinds of features are things that we can measure improvement in, hopefully pretty easily, and that was something that was important for us to look at carefully. It was actually an outgrowth of another observation that's been pretty well documented, which is that these kids have a lot of low tone, right? I mean, virtually all the children that we saw had low muscle tone. And so it might be that the gait disturbance is an outgrowth of the low muscle tone, but it's much harder to measure tone in a meaningful way and much easier to measure gait. And once we kind of talk a little bit more later on about the trial and some of the specifics, I can describe some details in terms of how we plan on measuring gait. So the other element that seemed to be very pervasive in meeting these kids was language delay, obviously. And that's been described in the literature, and that wasn't surprising at all. But how do you measure language? Historically, language is measured by querying parents. You say how many words, how many vocalizations. These are very imprecise instruments, and they're very difficult to see any meaningful change over time. Of course, we were seeing very significant language delays and language impairments in these kids, and we were trying to find ways of measuring it objectively. And there's been a lot of technology now dedicated to trying to measure language, and that's one of the things that we finally came across, which was essentially a digital language processor. So it's a chip that you can put in a child's vest or clothing that can measure language over however much time you want, 12 hours or even 1 hour, and it can do a very nice job of separating out between non-speech vocalizations, pre-speech vocalizations, or even speech. And so in terms of measuring improvement, it's the kind of thing where we're hoping it's going to be very sensitive and, of course, objective.
That's fascinating, and it's definitely going to have relevance to many different types of developmental disorders.
Exactly.
Dr. Buxbaum, coming back to the basic science, what does the high penetrance of autism and Phelan-McDermid syndrome mean for autism research?
Well, I think it means what I kind of intimated before, which is that this is a pathway that is likely to be impacted in many individuals with autism, right? And more, I would even go more broadly to say individuals with any kind of developmental delay. So I think that it seems very unlikely that all of developmental delay and certainly all of autism will resolve into a single pathway, but it seems likely that there will be a few main pathways that explain a lot of these sorts of syndromes. And I think that Shank 3 is going to be at the hub of one of the most important pathways.
Yeah, and so were there any findings from the clinic that you learned about that either challenged or confirmed your mouse observations?
Well, Geraldine, I'm actually a skeptic about mouse behavior as being the be-all and end-all of in terms of an outcome measure. I think we know even from the Fragile X mice, they show very, very little behaviorally. So I'm never particularly, at least when I don't see the gross changes in a mouse that you might see, I shouldn't say gross, I should say higher order changes like behavioral changes. I never really feel particularly challenged because I have low expectations there. But it is also true that the mouse models, no matter which ones you look at, the degree of social impairment is modest at best, and the degree of motor abnormalities is very modest at best, right? So, but they are there. So I'm happy to see them there. I'm not terribly surprised that they are not of a severity that you might see in PMS. Some of the things that we have yet to explore, the question about epilepsy. So the mice don't have obvious kind of epilepsy, although we all, everybody needs to go back and look carefully about, are they more susceptible to things that cause epilepsy? So it's not so much that we're challenged yet, but there are things from the clinical side that we need to circle back and examine the mice carefully and see how well they recapitulate those features, and then if they do, they become very useful models to understanding those aspects of the disorder.
Right. Absolutely. So about 1.5 years ago, we began to hear about a drug compound called IGF1 and its promise in your mouse model. Tell us what led to that discovery and what you observed in the mice.
So let me start with the last part. What we observed in the mice, as I said, was a pretty clear change in the communication between two nerve cells. And from a molecular perspective, it looked like that change could be called that the nerve cells were in a more immature state. So there's something called, you can take a ratio of two brain chemical signals, and you can that ratio changes as the nerve cells mature. And so one possibility was that ratio was low. So you could say that they were at a more immature state. We also did some morphological studies actually looking at the shape of nerve cells in the brain in the mice. And there too, it seemed to indicate a slightly less mature state of the synapse, or the point of contact between nerve cells. And that led us to think about factors like growth factors that might actually kind of enhance nerve development. I should also say that there are other things that we saw change that are also therapeutic targets, and we've tried some of those too, and they work just as well as IGF1. But the reason we put a lot of effort in IGF1 was partly, I gave you the rationale, that the nerve cells seem to be a little bit less mature, and IGF1 was known to promote maturity, but also IGF1 was already approved for clinical use and approved for use in kids. And there are very few drugs out there that actually are approved for use in kids that would have any properties that we would currently, based on our data to date, think of as being useful for therapeutics in either Phelan-McDermid or autism in terms of core symptoms, not in terms of many of the associated symptoms that we can actually do something about. So, people often ask me about the scientific rationale for IGF1, and I gave it to you, but I can tell you right now we'll have a stronger scientific rationale for drugs like ampakines. But ampakines are not available clinically yet, while IGF1 is. And so we spent a lot of time, the idea of repurposing existing drugs is one of the ways that many people are thinking about cutting short that drug development cycle that could be, a decade or more, depending on where you are entering the process. So for us, it became very exciting to think about an existing drug. And what we observed is that when you give IGF1 at a dose that's used already for a form of dwarfism to the mice for 2 weeks, and then you do the same assays I talked about before, which is the assays of brain nerve cell function and motor behaviors, we're able to reverse completely the things that were changed in the mouse model. So the mice then look like typically developing mice by those two measures. So of course it raises the possibility that some repeated exposure to IGF1 may bring the synapses to a state that's slightly more developed, and then kind of, if you will, normalizes the phenotype that you see in the mice, but presumably, hopefully also in individuals with PMS.
That's certainly our hope, and you're right. There is, I think, a real sense of urgency, and the idea, the rationale, or the idea of repurposing available drugs is one I think that can help accelerate this process for those of us who are desperately looking for ways that our children's lives may be improved. So that brings us back to Dr. Colazon. You're preparing to start a clinical trial of IGF1 in clinic. That's really exciting. This is going to be one of the first trials of a drug compound for an autism-related condition that emerged from studying mouse models. Tell us about the clinical trial.
Sure. So as Joe mentioned, I mean, we were pretty strategic in thinking about potential compounds. And when we selected IGF1 in the end and found this really convincing preclinical evidence, what we didn't anticipate was the associated costs. And as it turns out, this drug is something on the order of $40,000 a year, unfortunately. And so what that's required us to do, obviously, is to seek aggressively for funding. And Joe and I and a lot of people in our group put together a pretty comprehensive, what's called project grant around Shank 3, of which one component is a clinical trial. We submitted this grant for funding in November, and we expect to hear in the spring, but in June, right, Joe? June. So
Yes.
This large trial that we proposed is going to be with 36 patients. Every patient is going to receive IGF1 or placebo for 12 weeks. There's then going to be a 4-week period where the drug is washed out, and then people are going to switch over to the other treatment group. And what that allows us to do is essentially use every patient as their own control, but it also ensures that everybody who's participating is going to be able to get active medication. The challenge around the finances of it obviously require that we get full funding to proceed. In the meantime, though, we've been able to collect enough internal funding and through other sources to begin a small pilot trial now in just 10 patients. And we're actually very happy to report that as of Monday this week, our internal review board at Mount Sinai has given us approval to start this small trial with 10 patients. And the smaller trial is going to also follow the same design, where there's going to be treatment for 12 weeks, a washout period, and then a switch into either active treatment or placebo in the same way. I know that a lot of questions have come up. Would you like to ask me each question, Geraldine? You want me just to anticipate them?
Okay, so you did mention that the trial would be starting soon. When do you anticipate it would start? When do you anticipate the pilot trial would start?
So safety is really our primary concern here, right? This is a totally new compound, and even though it's been approved by the FDA for use in children with IGF1 deficiency, we really have no idea what the safety profile is going to look like in children with Phelan-McDermid syndrome. And so there's a number, now that we have approval to proceed, there's a number of pieces that we need to put in place between now and hopefully a month from now. And I hope to be able to begin within a month, frankly. We were trying to figure out what a reasonable start date would be, and we were projecting January. And so we're not that far off, but a month from now, I hope to be able to enroll our first patient.
You mentioned you need to consider some safety issues. Why don't you describe the inclusion and exclusion criteria and sort of the rationale for these criteria, that is, who can be involved in this study?
Sure. So IGF1 is a growth factor, as Joe mentioned, and our knowledge about it is really based on long-term treatment in children with short stature or IGF1 deficiency. And because it also acts along this growth hormone insulin pathway, it can really reduce blood sugar. So the two things that I'm most concerned about is inducing growth, where we don't want growth to occur, except in the brain, obviously. That's where we want the growth to occur. But I'm talking about growth in bones, for example. And we also don't want to put kids at risk of significant hypoglycemia. So very low levels of blood sugar can be associated with a lot of different kinds of symptoms, dizziness and sweating and palpitations and that kind of stuff. But it can also be associated with an increased risk of seizures or even potentially unconsciousness, right? So those are the two factors that have kind of driven most of our discussion around safety. In the end, what we've decided was to really only include children who were 5 years old and older, and up until children who have completed their growth. So after a certain age, and it's different for every child, your growth plates fuse in your bones. And if you give a growth factor to someone whose growth plates have fused, it can be quite dangerous. So those kids we absolutely have to exclude. So anybody over 17, unfortunately, we're going to have to exclude, and anybody under 17 whose growth plates have fused, we're going to have to exclude. At the same time, although the drug has been approved in kids younger than 5, down to actually 2 years old, we really wanted to be much more careful in this first attempt, and we wanted to learn more about the drug before we start thinking about using it in kids between the ages of 2 and 5. The other piece of that calculation is actually a scientific one and a strategic one, where many of the outcome measures that we're using, particularly the behavioral outcome measures, really haven't been as well validated in children under the age of 5. And because so much of this hinges on funding, we wanted to be pretty conservative in terms of what we were proposing, and we didn't want to raise concerns among reviewers of this grant, for example, that we were being careless or that we were potentially using outcome measures that would not pick up change in kids under the age of 5.
Right, right. Tell me about where the study is going to be conducted and which participants can be included for that geographical area.
Right. So the initial studies, both this small pilot with 10 kids and hopefully the larger study, if it in fact gets funded, of 36 kids, it's going to occur here at our center. Our goal, of course, is that the moment we can actually see some benefit, the moment that we have actual evidence of benefit and feel comfortable with the safety profile, we're going to immediately submit those preliminary results for a grant to propose a multi-center trial. You know, the goal is obviously to include as many families as possible, and we do have collaborators all around the country that I'm sure will be interested, and several of whom have already expressed clear interest. But because of these safety concerns and because of the fact that we're using some behavioral outcome measures, we've restricted the inclusion to include only people that are really within travel distance to the center. The people that have to be able to kind of comfortably come to our center every 2 to 4 weeks for safety and efficacy assessments. I know that there have been so many wonderful families that are unbelievably committed to this and have poured tons and tons of energy into it and would frankly relocate. They'd move to New York, and we get that, and we would love to include them. The issue for us is in that case something of a scientific one, where if you take a child out of their school and out of their home environment, and if you interrupt their other behavioral therapies and speech and language therapies, then we're bringing them here to New York and giving them a medicine and trying to measure improvement across language and behavior, for example, and it becomes a real significant confound. And so what we've done for this kind of initial pilot is created, we've got, I think, 50 or 60 people who have called us and expressed interest, and I've asked somebody to input their address and set up basically a graph of how far people are that are interested from our center by miles. And I'm just going to go down that list, essentially.
Okay. Okay. So you mentioned that IGF-1 can be a very risky drug because of its effects on growth and on kind of the insulin pathway. I think for a lot of families who are dealing with some really challenging issues with their children, it's going to be tempting to try to get IGF-1 some way or another. Tell us about the risks of doing that and why that's not a good idea.
Yeah, thank you for bringing that up. So it's worth spending a minute or so just on describing some of the risks, because I guess it's not something to take lightly. There are some common side effects of IGF-1, and we mentioned one of them, low blood sugar. Other ones can include increase in liver function, elevated cholesterol, triglycerides. Kids can have, this is less common but can still happen, they can have enlarged tonsils. There can be increased pressure in the brain. There can be hearing problems because of the increased pressure. You can have headaches and, heart murmurs, and there's a lot of assessment that's going to go in initially to making sure kids are eligible and it's safe to study them. We're going to be doing EKGs and echocardiograms, and we're going to be doing neurological exams, and as I said, a lot of assessments. So, people ask me all the time about, can I use this experimental treatment? And my perspective is always really around the evidence. And at the moment, there's absolutely no evidence to really support the use of this medicine clinically without studying it in some controlled way. And so I think that even though I'm generally in favor of using medicines to gain experience and to see if there's potentially benefit, I think here we really can't do a fair risk-benefit calculation because the risks are clear and the benefits are not known. So I really caution people not to try to use this off-label. And then the other piece of it is just, it's again back to this idea of kind of scientific rigor. If people in this very small community start using this drug, and we want to do larger and larger, more definitive clinical trials, and we want to eventually, let's say if we see some positive results, go to the FDA and get a label and have a drug company actually give it to us so that it's covered by insurance. All these issues mean that our methods have to be clear. And if people are on drug, it's going to be impossible for the most part to have them participate in the trial.
Right.
And so I just think that if it's going to be used, it should be used in controlled conditions.
Right. I think that's a point we definitely need to emphasize. Going back to the potential side effects, that's one of the reasons, right, that you'll be seeing children or the patients in clinic every 2 weeks in order to monitor all of these potential side effects.
That's exactly right. The first 4 weeks of the 12-week period is going to be an increased titration, where we're going to be slowly increasing the dose. And during that period, we're going to be talking probably several times a week, and we're going to be seeing kids every other week for blood work and for examinations. After that, it's going to be every 4 weeks, but it is it is a big time commitment for sure.
Right. Who should families contact to learn more?
So we have a wonderful coordinator. I think a lot of families have got to know our coordinator, Sarah, who's since moved on to graduate school. But we have a coordinator named Jessica. Her last name is Zwefach, Z-W-E-F-I-C-H, and her phone number is 212-241-28. Oh, look at that! It's on the screen. 2826. And you can put my information up there also. A lot of times Jessica is very helpful in answering logistical questions and has had now a fair amount of experience putting together this grant and knowing what we're doing. But people are also certainly welcome to call me directly, and often Jessica will help coordinate a time where she knows that I'll be available, and so we don't have to play too much phone tag.
So going back to IGF1 and this clinical trial, what do you think might be some of the possible outcomes of this clinical trial?
It's a great question, and really the only answer is I don't know. What you do in these kind of early phase studies is you pick lots and lots and lots of outcomes, and you try to cover as much base as you can. So we're interested in language outcomes. We're interested in social outcomes. We're interested in repetitive behavior outcomes. We're interested in motor skills outcomes. We're interested in cognitive outcomes, learning and memory paradigms. Those are all things that we're going to be measuring, and I just don't know. I don't know what we're going to see. The primary outcome that we've sort of selected on more of a strategic level than anything else is social withdrawal, because this is, as we see it, a core deficit in autism. Again, 80% of these kids are sitting somewhere on the autism spectrum, and we think that this particular outcome is going to be high yield, and it's also one that's very familiar to the autism community, and we have a very well validated instrument to measure it.
Right. Well, we wish you the very best with your clinical trial.
Thank you. Thank you.
Dr. Buxbaum, the scientific community is just out of the gates in the field of Phelan-McDermid syndrome and autism research. What are some of your current and future projects?
So, have I unmuted successfully? Let me start with that.
Yes, we can hear you.
Oh, good. So, we are continuing to dissect some of the changes that are in the mouse models. We're also developing additional mouse and rat models. We are doing a lot of work on there are various high throughput methodologies to ask what's changing in a brain. So we're doing what's called two-dimensional gel electrophoresis. We're doing large-scale RNA experiments. So we're basically using what are called genomic methodologies to survey to kind of get a sense of what's changing that we wouldn't necessarily predict would be changing. Because a lot of times in science, you look under the lamppost, but there are now methods to look in an unbiased way everywhere, completely unexpected. So we're doing that. And kind of to circle back maybe about the question of IGF1 and its, that it does have a restricted age range, and it is maybe, it does have some risk associated with it. We do think, and we're moving a little slower than we'd like, but we're moving in a direction where we're trying to understand the mechanisms by which IGF1 works, because then we can think about other drugs that might be more tractable, small molecules, or that can be used over a wider age range that can actually do the same thing as IGF1.
Right. So important. So you mentioned your Shank 3 knockout rat. That's really exciting. It's really going to be one of the first rat models of autism. How do you expect the rat might be different from mice, and is there anything you can share so far about your preliminary findings in the rat?
Sure. So it's kind of silly to say, but rats are bigger. And that turns out to be pretty important for studying neurobiology. It's much easier to put an electrode in a certain part of brain tissue in the rat versus the mouse. The cells themselves are significantly bigger, so you can do more with them. So some of the most basic neurobiology is actually much easier in rats. Also, rats have a much more elaborated behavioral repertoire and much more complex social interactions than mice. Even though mice are perhaps the more social species in terms of living in social groups, rats have a much more complicated repertoire. So, there's good reason that there's unquestionably it's going to be easier to do neuroscience in rats, and there may even be better behavioral assays we can do in the rats. So that, and I will just say, just in terms of drug development, when pharmaceutical companies try to start on toxicology, trying to understand how drugs are metabolized, they work in rats, not in mice, as a first step. So,
wow. Okay.
So if a pharmaceutical company is confronted with data in mice, and then they have to kind of flip over to rat, able to, because they don't have the rat with the knockout. Right? So they learn something from a mouse, and they go to a rat to do the pharmacology, metabolism, and toxicity, and then they go on to additional model systems. Here, the discovery and the discoveries about what drugs might work will be done in the same organism that they would do the first line tox and metabolism screens. So that's, I think, that's going to be a very important shift in moving things from the very preclinical space to the more kind of proximal clinical space. Mm-hmm. And in terms of preliminary rat findings, what we've done first and foremost is kind of make a checklist of what we see in the mice in terms of electrophysiology and see if we see the same things in the rats. And so far the answer is we do. We see deficits in long-term potentiation. We see deficits in the connection between nerve cells and in the plasticity or change of those connections. And as uninteresting as that sounds, because all we're saying is we're seeing the same things we saw in the mice, it's an important comparator study to do. Mm-hmm. Now that we know that the rats look, at least by those measures, pretty similar to the mice, we've just started now going into additional brain regions, particularly what we call the prefrontal cortex, which is involved in a lot to do with emotional regulation, with language, with cognition. And it's a brain region that's very tractable in rats and very difficult to study in mice. So we've actually made that jump, and we're able to actually study the activity of nerve cells in the living brain in these frontal areas. And we think that that's really going to be a region that's going to provide deep insights into not only PMS but also autism more broadly.
Right.
So that's what we're working on.
Yeah, that's exciting, Mark. Good luck.
Thank you.
Wish you the best. So we're actually going to field some questions from the attendees now. Sue has been receiving questions through the messaging system on GoToWebinar, and I'm going to turn it over to her now.
Okay, thank you, Geraldine. We have a couple questions about IGF-1. The first is, Dr. Collison, are you going to use the same IGF-1 that is currently being tested on people with Rett syndrome, and if yes, how?
The answer is yes. We're using the commercially available form. That's the one that the FDA has approved for us to use. It is the same one that the group in Boston that's studying Rett syndrome is using. And it's a subcutaneous formulation where it's delivered under the skin in the same way that insulin is delivered with diabetes. And it's a dose that's given twice a day. And we'll be giving it there's a pretty clear dosing guideline that is published in the package insert, and we'll be following that to some extent on a slightly more conservative basis.
Good, thank you. Dr. Buxbaum, this is another question we received prior to the webinar. I have read a number of studies where IGF-1 was successfully administered intranasally in both mice and rats. Has the Seaver Center tested intranasal IGF-1 in their knockout mice, and if not, would they consider doing so?
So some time ago we began to work with some intranasal administration, and we actually think it works, but that is not a again, we're going to an area of clinical trials using approved drug and also approved routes of administration. I'm not sure it would do the families much benefit to have to go through approval to get an addition to get approval to try a novel route of administration.
The follow-up question with the intranasal IGF-1 is, could it be possible to avoid many of the growth-related side effects while maintaining its brain development benefits using the intranasal IGF-1?
So I mean, I think Alex may answer this better, but I don't think there's evidence of people growing too much if they take IGF-1 if they don't have short stature. But again, let me just say what I said before, that any change in administration would require a full IND, safety, efficacy as well. Alex, want to take that from me?
Yeah. So part of your response was broken up, Joe. Jesse's sending you a message on the board, but so there's a couple of issues. One is, again, this idea that IGF-1 has primarily been looked at for long-term treatment in only people with IGF deficiency and short stature. I don't anticipate a lot of growth in a 3-month trial. And whether or not using an intranasal formulation that's delivered just more specifically to the brain is going to, in some ways, mitigate that risk, it would just be speculative. I don't really know.
Okay.
But Alex, what would you need to do to get approval to try?
I can't, Joe. I'm sorry. I can't hear you.
Yeah, sorry, Joe. We can't hear you anymore. So I'm just going to direct to Dr. Collison right now, if that's okay.
Sure.
Dr. Collison, are the upcoming trials going to be limited to PMS patients that have been duly diagnosed with autism?
Great question. The answer is no. No, we thought a lot about that, and in the end decided to include all patients with pathogenic deletions or mutations in Shank 3.
Great, thank you. I do have a question here. Are you collaborating at all with Children's Hospital of Boston researchers?
So we're collaborating closely with them in the sense that Dr. Khwaja has agreed to be a consultant on our grant and has already agreed to share a lot of his protocol and his safety data and his titration schedule. So he's been here to talk, and he and I have met, and I'm constantly hammering him with emails about his experience with IGF-1.
Good. I have a question. You've already talked about this, but could you explain again the risks of obtaining IGF-1 off-label and what it would do to the process?
So it's a very small community, obviously, and putting aside the financial burden, right? I mean, assuming that it was easily accessible and it didn't cost so much money, if lots and lots of people started to use IGF-1, it would potentially confound our ability to recruit patients for clinical trials. And so if phase 1 is 10 people and phase 2 is a 36-person trial and phase 3 is a multi-center trial, and that's the typical pathway that one follows to try to get approvals for drugs, that pathway is going to be compromised if there are many people in the community that are using it on their own and basically not participating in trials. That was my primary concern. But no, my primary concern is that they're going to be using it without any evidence of benefit and without sufficient monitoring in terms of safety. But then the secondary concern, of course, is just in terms of this more strategic issue.
Thank you. Geraldine, do you have any more questions? Because that is all I have right now.
Let me just check my list here. Oh, one other question. For if the center grant is funded, have you identified who your other collaborators might be and what? Or I'm sorry, if you move to a third phase clinical trial, have you identified what other centers might be your collaborators and where the trials might occur?
So it's really very preliminary, and we've had preliminary discussions with people on the West Coast in Washington and also Midwest and Chicago, and we have lots of friends and people that we have a long history of collaborating with in the South and really all over the country, because even though we've not been enormously successful in terms of bringing effective drugs to bear on communities and autism in general, we have been pretty successful at creating clinical trial networks. And so the infrastructure already exists, and the expertise is there. So I don't anticipate it to be an issue, as long as the funding is in place and as long as we have some preliminary evidence of benefit. But you know, we'll be as inclusive as possible. 5 sites is not an unusual sort of number of network sites. And then, you know, if depending on how definitive a trial you want to do, people can do sometimes 12 sites or even more. Especially in rare disorders, it might be necessary, right?
Okay, very good. Thank you. I think that was it for my list of questions.
Okay. Well, really special thanks to Drs. Collison and Buxbaum for participating in our first ever webinar. We wish you continued success in your research. Thanks to everyone for joining us today. At the conclusion of the webinar, you'll be directed to a survey. Please take a few moments to provide some feedback so we can learn how to improve the webinar experience. Thank you so much, everybody.
Thank you. Alex and Joe, thank you so much for participating. I really appreciate your time today and your participation.
Sure, it was really good to be here. Thanks for inviting us.
Thank you, guys.
Okay, talk to you all later.
Okay.
Bye.
Bye.